Continuing our discussion of mechanical imaging modes of the AFM, this next slide shows some examples of images. Panel A is contact mode AFM, done in rather special circumstances. It's a very stable layer of membrane protein channels absorbed onto a mica surface and imaged in a salt solution chosen so as to minimize adhesion between the probe and the sample. Also, a very sharp probe was used. One can see every domain of the protein. There are actually 6 of them, arranged like this. And as the probe is scanned upward in the image, the contact force is increased, and you see at the top part of the image a dramatic change in conformation of the protein as 3 domains pop out of the surface as a result of the large force exerted by the probe. B is an example of a dynamic force microscopy image. What you're looking at here is a chromatin, which is a combination of DNA, so the little spaghetti-shaped molecule is DNA, wrapped around a group of positively charged proteins called histone proteins. These form nucleosomes, which then fold to form chromatin, of which chromosomes inside cells are made. This is an image taken in solution, and you can see that the resolution is very high. You can see the DNA quite clearly. This was actually taken by dynamic force microscopy with a magnetically oscillated probe, and we'll come to that in the next slide. Panel C shows non-contact imaging. What was done here was a set of stripes of water were absorbed on the surface. So you're actually looking at a surface here with water stripes on it like this. This was done by making a surface hydrophilic in certain areas and then exposing it to water vapor. The non-contact mode is evident because the soft surface of water can be imaged quite clearly. However, when the amplitude is increased to start to bring the tip closer to the surface, you see in the last stripe over here the instability in the image as the probe begins to disturb the water on the surface. Panel D is an example of this wonderful FM-AFM mode of imaging. It's of the silicon 7x7 surface. You probably recognize this diamond-shaped lattice from the STM image taken by Binnig and Rohrer that won them the Nobel Prize. Only in this case it's taken with an atomic force microscope. It's a very flat surface prepared ultra clean in ultra high vacuum, and the sensitivity comes here from the fact that in vacuum the probe can be made so as to oscillate at many megahertz in frequency, and by dwelling the probe over the surface for long enough one can measure to a very tiny fraction of a hertz, resulting in this absolutely glorious atomic scale resolution of the silicon 7x7 surface. Let's turn now to a variant of dynamic force microscopy developed for imaging in fluids. This is magnetic AC or MAC mode, and it operates the following way. So in operating in fluids, one has the problem that the cantilever has to push against a large mass of water in order to oscillate. And for that reason, the oscillation applied to the piezoelectric stack to drive this piezo into oscillation has to be very large indeed. It's a bit like trying to make a lettuce leaf wiggle in a bathtub. So this means that when you set up enough drive to drive the cantilever into a bending mode resonance, rather than having this nice plot of amplitude against frequency that I'd sketched out for you earlier, you typically see something very noisy with many peaks in it, and it's hard to decide what the correct resonance is, and the resulting image is somewhat noisy. In MAC mode, this problem is solved by putting a magnetic film on the cantilever and making that the only magnetic component in the microscope, and then under the sample having a solenoid that generates an AC magnetic field that acts only on this cantilever, moving it directly and therefore succeeding in getting a reasonable drive in solution without shaking the whole microscope. The resolution can be very high indeed. These images here are of tiny DNA microcircles. They were made by forming closed circles of DNA of just 168 base pairs. The diameter of the circles that you're looking at here is only about 20 nanometers across the whole circle. And the structure that you see going around the circle in panel A is in fact owing to the helical turns of the DNA double helix. Because this experiment was, this measurement was made in solution, it was possible to change the ions in solution and observe this rather remarkable effect that when you change from magnesium to zinc, the DNA forms kinked squares rather than smoothly bent circles.